as per our formulation, as per our equation, we have to apply the sometimes sometimes homotopy perturbation method is good, sometimes homotopy analysis method is good, sometimes. So depending on the your example, or depending on the your equation, we are using.
Okay, sir. Thank you, sir. Very nice reply, sir.
Thank you.
Already we used this type of all methods, sir. Various vibration method, homotopy analysis method, homotopy perturbation method, all are already we used.
Yes,
When
yes.
compared to the numerical solution, Heller box method,
Hmm.
Galerkin method, finite element
Hmm.
method, finite volume method, all are compared, sir.
Hmm.
But some problems we don't get the analytical solution, sir.
Yes, yes.
Even perturbation techniques also first two term only we will get, sir.
Yes, yes.
Another
That's
terms it's very difficult.
so.
So that is
For that, I can suggest you that you try to neglect some non-linear term, find out the exact solution. Okay, and
yes, sir.
then you can also apply the numerical techniques for your equation, find out the solution, and whatever the terms you neglected, okay, same things you can use in the numerical simulation and compare your result. So that type of validation we are doing.
Yes, sir.
Now you
Yes, sir.
got my point.
Yes, sir. Yes, sir. Got it, sir. Got it.
Okay,
Thank you, sir.
thank you, thank you.
Thank you so much.
Is any more query? Hello.
Madam Nasrin, madam, any other questions from audience?
No, I think, sir. Thank you, Dharmendra Tripathi ji, sir. Thank you so much.
Thank you for inviting, and thank you
sir.
for giving such opportunity to interact with you people. Thank you very much. So I'm leaving because I have some other work. Thank you.
Sir. Thank you so much, sir.
Thank you.
Sir, shall we start presentation session, sir?
The main today's topic is synthesis, chemical, electrochemical, and biological characterization of metal complexes. So first, let me explain what is a transition metal complex. Sites, that is denticity is a 4, either which will undergo the tetrahedral structure or which will undergo the square planar structure. So depending upon the nature of the metal, what type of hybridization it undergoes, so accordingly which will undergo any one type of geometry or the structure. If it is 5 coordination bond, then generally which will form a trigonal bipyramidal type of structure. So otherwise, if the coordination bond is or the coordination sites are for the ligands, the denticity, if it is a 6, which will undergo the octahedral type of structure. So generally when we try to form a complex between the metal and the ligand, so mostly either which will undergo the 4 coordination type or 6 coordination type. So these are the two the most commonly involving the coordination complexes. So either which will form a tetrahedral or square planar structure, otherwise octahedral structure. So to form an octahedral structure, the minimum requirement is one metal ion and the ligand having the 6 coordination sites. So similarly, depending upon the coordination sites which will undergo the coordination with the metal complexes. So next, let me explain the that is synthesis, then the various properties, how to characterize with the case study with one simple example. Say for example, I have chosen the ruthenium complexes. So why I have chosen the ruthenium complexes? Actually, this ruthenium complexes is very easy to handle. Very easy to handle in the sense, whatever may be the applications we want, accordingly we can mold the complex, we can prepare the complex. So, and at the same time, one more disadvantage of this ruthenium complexes is which is highly unstable, and at the same time, which is costlier also. So actually the weight of the ruthenium, that is ruthenium, mostly the basic compound we'll be buying in the form of ruthenium trichloride, and its cost is costlier than the gold. So that's what the disadvantages. Otherwise, if we handle this metal, it is very easy to mold. Whatever may be the ligand and whatever may be the geometry we want to arrive, it is very easy to arrive, and at the same time, which will find many biological applications. So not only the biological applications, one more, the major advantage of using this ruthenium metal is which will be having variable oxidation states. So because of this variable oxidation states, it is very flexible to undergo the oxidation and reduction in that way, which finds many applications in the redox properties. So because of this flexible redox properties, it finds applications in the catalytic studies, and this catalytic transformations finds applications in the pharmaceutical industries for the various functional group transformations, say for example oxidation, hydrogenation, or reduction, or hydroformylation. So for this type of reactions, we can use this ruthenium complexes. So which is mainly because of its flexible oxidation states or varying oxidation states. So most of the ruthenium complexes which finds applications in the cancer therapy. Whatever may be the ruthenium complexes, if we try to synthesize, generally which will be having some biological properties, and definitely which will be having cancer, that is anti-cancer property also. So to find out the anti-cancer properties, there are so many methods. Let me explain one by one in detail, and after explaining the synthesis. So let me share the synthesis process. So actually the same compound, the same ruthenium-3 complexes. So now all the documents I am downloading from the patent office site only. The same complex we have prepared, we have characterized properly, and we have proved its anti-cancer potential, and finally we filed a patents, and we received a grant also. I'll show you the certificate and how to process and how we have defended to get the patent grant. And this is what the disclosures we have submitted at the patent office. So to synthesize this complex, so initially we have to synthesize the ligand. So the ligand we have synthesized here, it is, so I think you can able to view. Yeah, this is what the experimental session. Yeah, initially, actually, this complex is synthesized by 3 steps. So, generally, whatever may be the complex, which will be synthesized by 3 steps, sometimes which may extend one more step for the synthesis of complexes, depending upon the oxidation state in which we are going to arrive. So, initially, we have to decide that whether. So mostly the ruthenium complexes, among the various oxidation states of ruthenium, ruthenium-2 and ruthenium-3 is most stable. So we have to, even among the ruthenium-2 and ruthenium-3 is still more stable. So we have to prepare the ruthenium 3 straight away. Otherwise, if we want to prepare the ruthenium 2 complexes, so since it is not that much stable, so always we have to prepare it in the inert atmosphere. So, and at the same time, we have to prepare in the restricted atmosphere, that is by purging the inert gases like nitrogen. In the nitrogen atmosphere only we have to try to synthesize. Otherwise, if we try to find out the applications in the ruthenium 3, then it is very easy to handle while synthesis itself. So, the synthesis process contains 3 steps. One is the synthesis of ligands. So, that's what the first step. So here the 4-fluoro-phenyl-iminomethyl and then the substituted phenol is synthesized. So by adding the, this is a type of Chif base. So that's what I wanted to convey. And for this ruthenium complexes, the most convenient ligand is Chif bases. So Chif bases contains its amine group and CO group, amine group and CO group. So generally Chif bases are obtained by the condensation of this aldehyde, and either it may be aldehyde or ketone. It has to have a C double bond O and NH2 group. So in that way we have taken the 2-hydroxybenzaldehyde, which contains the aldehyde group, and this 4-fluoroaniline, which contains the amine group. So both are taken in terms of 1:1 molar ratio, and which will be dissolved in methanol of 25 cc, and the solution was heated under reflux for 3 hours with a continuous stirring, and finally which will be concentrated to 5 ml, and which is upon cooling. So there is a yellow-orange crystalline product was separated out, and which is washed with the cold ethanol and dried, and the same is recrystallized by using the same cold ethanol. So this is what the scheme of the synthesis of the ligand, and this is what the amine part, and this is aldehyde part, and we have to do the reflection process for 3 hours in the presence of methanol. Finally we will be arriving the ligand. And the next scheme is the second step of the synthesis of metal complexes is synthesis of ruthenium starting complexes. So except the ruthenium, the all other complexes like copper, cobalt, nickel, zinc, then oxovanadium, even manganese, magnesium. So these metal complexes can be prepared directly by reacting any one of the salts of the metal with the Chif bases, that is ligands. But to synthesize this ruthenium, so we have to have 2 steps. The first step is the synthesis of starting complexes. So if we do this by 2 steps, the complex will be very stable, and so anytime after completing the first step, the second step of the synthesis will be completed. And how to prepare the ruthenium starting complexes? To prepare the ruthenium complexes, as I said, we have to start from the commercial ruthenium trichloride, and so it has to be reacted with the Chif bases. So always ruthenium complexes forms the octahedral structure. To form the octahedral structure, so it has to have 6 coordination sites. So the Chif bases generally have 2 coordination sites. If it involves only one Chif base in the complex formation, which contributes 2 coordination sites, which will be satisfying the 2 valencies of the ruthenium, and the remaining 4 valencies will be satisfied with the co-ligands. Here the co-ligands I have chosen is 1,10-phenolphthalein and bipyridine. And for only one compound I have chosen the diphenylphenazine also, and it is a little bit difficult to handle since which will be soluble only in DMSO, dimethyl sulfoxide, which is highly carcinogenic, and we have to be very careful, and we have to handle very carefully. So otherwise the other co-ligands which will be dissolved in DMF, dimethyl formamide. To form this complex, again we have to do the reflection process between the commercially available ruthenium trichloride and the co-ligands for 8 hours, and then the reaction mixture will be cooled to the room temperature, and which will be taken out and which will be added with the acetone solution, and finally which will be kept at 0 degree. So mostly which will be kept inside the freezer for overnight, and then the next day if we see the crystals or the products will be formed, and the product formed which is in the microcrystalline nature and which will be washed out thoroughly and which will be dried using the desiccator. Sometimes using the same process we can prepare the crystalline substances also. The same ligand we have crystallized and we have characterized with a single crystal XRD, and we have published the same in the Acta Crystallographica section E. And the third step of the complex preparation is so again the starting complex. So the starting complex just now we have prepared by the second step, again which will be dissolved in ethanol and silver nitrate, again which will be stirred with the ligands, and that is by the time what will happen, this silver chloride will be separated and which will be removed, and then which will be added with the this sodium salt of alcohol, and which will undergo the reflection for 1 hour, and this reflection will be taking place in the presence of water bath. There are two types of reflection. So directly by using the heating mantle we can do the that is this process. Otherwise the second type of this one is in the presence of a water bath, and here we need not to follow the temperature. So we have to say whatever the distillation temperature, that temperature is enough. That is one of the advantages. So generally if you see, the process will be taking place either at elevated temperature or at low temperature. Here one of the advantages as such, so whatever we are applying the temperature for the reflection process, with that temperature we can continue. And so the saturated aqueous solution of sodium chlorate is added, and finally the precipitate is separated, and which will be settled and which will be filtered by using the crucible. And finally this precipitate is washed with the cold water and then dried in the presence of anhydrous calcium chloride. And the same thing can be recrystallized by using the slow evaporation process. So this crystallization is done by using the 1:1 acetonitrile and benzene as the solvent, and finally we got the dark red crystals of the complex. So okay, the complex here it is RuAbFpImp. That is this is nothing but the Schiff base. We have differentiated with the two substitutions. So okay, the substitution X and Y, it may be hydrogen or halogens like chlorine, bromine and iodine, and ClO4.2H2O. Here the yield obtained is so 82 to 86%. Here A and B are the co-ligands. Either it may be 1,10-phenanthroline or bipyridyl or diphenylphenazine. So in the same way we have prepared the other complex also. So here in this formula 2 synthesis only there is a slight difference. So from the diagram we can understand. So this is what the starting complex and this is what the ligand. So we have synthesized by using the first step, and this one we have synthesized by using the second step, and the third step we have synthesized by using this scheme, this step. So okay, and finally we will be getting the ruthenium-2 complex, but our aim is to get the ruthenium-3 complexes. For that we have extended one more step. So that's what the synthesis of ruthenium-3 complexes. So likewise we can synthesize all the substituted complexes. So next thing, we have characterized using the spectral studies, spectral characterization. So actually for the patent filing, this spectral characterization we need to submit as a separate file, as the additional file, that's what which is not included in the drafting. So we have, for the characterization of ligand, we have used UV visible spectrophotometry, then infrared spectrophotometry, and then NMR, magnetic resonance spectrophotometer. So by using these spectral studies, we have concluded the structure of CHIF base as we showed earlier. So this is what the structure of CHIF base. And to characterize these complexes, we have used UV, IR, then ESIR, and then CV, cyclic voltammetry. So this ESR spectrophotometer is to find out whether which will be having the paramagnetic electron or not. So if it contains paramagnetic, whether it is paramagnetic or diamagnetic, accordingly its magnetic property will be. So to check that, we have characterized by using the ESR spectrophotometry. And finally we have run the CV, cyclic voltammetry also, and this is one of the most important characterization of especially this ruthenium complexes. So in which oxidation state it is existing presently, and what are all its variable oxidation state, to understand that based on that we can predict what will be the catalytic property. So in that way we have characterized this cyclic voltammetry. And from the electronic characterization we can understand that so what will be the number of unpaired electrons. So from the unpaired electrons also we can understand the magnetic properties of the compounds. And next is biological characterization, and this is the most important part of this complexes. For the biological characterization, actually for these complexes we have run the biological characterization for antibacterial, antifungal, even by theoretically we have found out anti-osteoporosis, and finally we have concentrated the anti-cancer properties of this complex, and this complex is having very good anti-cancer property that we have tested in vitro. So actually in vivo also we can characterize this complex biologically, at what extent it will be effective for anti-cancer properties. But we have to get a ethical clearance and all, since we are not having the biological lab by the time. So we cannot do the study using the in vivo studies. That's what, by using the cell lines we have done the in vitro type of evaluation to find out the therapeutic effect of the synthesized complexes. And the protocol used is the compounds are tested on 24 well trays, and cells are allowed in a flask. The grown-up cells are harvested just before they became hemocycometer, and then diluted down with media to a concentration of 10 to the power 4 cells per ml. Then the cells are then seeded in the 24 well trays at a density of 10 to the power 4 cells per well. Actually this is a common procedure only, but so whether, see, by trial and error we have to check whether this method is working out or not, or accordingly we have to change the density or the counts we have to change, and this we can optimize only by trial and error. And the same cells are then left to play down and grow for 72 hours, and before adding the ruthenium compound. And then the ruthenium complexes were weighed accurately and made up to the concentration of 1 milligram per ml with 1% of dimethyl sulfoxide. Then the appropriate volume of ruthenium complex solution is added to 5 ml of media to make it up a concentration of 100 micromolar to each drug. And this 100 micromolar solution is then serially diluted to make up to 10 micromolar. then 1 micromolar and 0.1 micromolar solutions. Accordingly, we have to step up with various dilutions, and for the various dilutions we have to find out at which dilution the compound will be effective, and based on that we can predict the effectiveness of the compound. And each concentration is done in duplicate in order to check the in order to ensure whether the results are correct or not. To ensure that we have run once again and all the results we have duplicated to check whether the results are correct or not. And finally the cells are left exposed to the drugs for 24 hours and then washed with buffer, in particular phosphate buffered saline, before fresh media is added, and they are allowed to grow on for a further 3 days before being counted using a counter. So using this counter we have to find out finally what will be the count, and based on that we can come to the conclusion that at what extent which will be effective. This is what the procedure to do the in vitro studies, and then how the cells are so that is the preparing the cells for count. So for that also there is a procedure. So based on that we have counted, and finally we have evaluated for the cytotoxic evaluation also. So this cytotoxic evaluation is to understand about the toxicity. This toxicity is nothing but the side effects when we use these complexes as the drugs. So to understand the side effects we have to do the cytotoxic evaluation. So for that also there is a procedure according to 3D chelation theory. So generally this transition metal complexes will be having less toxicity than the other organic compounds. So other organic compounds, so many organic compounds are synthesized and proved as anti-cancer agents. So instead of the organic compounds, if we use these metal complexes as the anti-cancer agents according to 3D chelation theory, which will be having less side effects and at the same time which will be more effective, it can able to penetrate into the that is cells which is affected by the cancer cells. So finally we arrived at a cytotoxic effect. The cytotoxic effect will be that is the results of side effects. That is cytotoxic effect will be expressed in terms of IC50 values. So here these values, lesser the values, higher will be the or more effective of the that is cytotoxic effect, is otherwise known as side effects. So here we have arrived up to 0.27. So these complexes are not having much side effects. That's also we have proved, and this is what the that is tabulated or documented cytotoxic values of the various compounds we have synthesized. Even we have screened the same thing for the ligands also. So this many ligands we have prepared, totally 8 ligands we have prepared, and we have prepared the complexes up to 18 complexes. For all the complexes we have tested the anti-cancer activity and at the same time cytotoxic effect, and from this we can understand that almost all the complexes are having very less cytotoxic effect. Among the various substitutions, this chloro-bromo substitution is more effective in terms of cancer fighting, and at the same time which will be having very less side effects. So here the compounds are extended for extended by varying its substitutions. So totally we have prepared 40 compounds. And the next one is DNA binding experiment. So this experiment is the evidence for the proving that so these complexes are very good anti-cancer agents. So this DNA binding experiment is done by absorption spectral titration, and this is done by using the UV visible spectrophotometer. So for this what we have to do, we have to we have to allow to interact these complexes with the DNA. For that we have to carry out this test by using the tris buffer. So this tris buffer is made by using 5 millimolar tris and 50 millimolar sodium chloride, so that the pH will be maintained almost at neutral pH. 7 is the neutral pH, so which will be 7.2. With this pH only, this DNA will be original or active as such. Otherwise, if is there any changes in the pH which will be denatured, so the results may not be correct. So we have to maintain the pH. With that pH, we have to do this absorption spectral titration, and during this absorption spectral titration, so we have to vary the concentration of I think so the graph is not here, so we have to vary the concentration of here so there is a concentration of this nucleic acid. Nucleic acid is nothing but the DNA. So we have to fix the concentration of metal complexes as 2 micromolar, and by varying concentration of this readily available DNA nucleic acid. So here we have used 2 types of DNA, that is half thymus DNA and this herringbone DNA, and we have varied from 0 to 20 micromolar, and for all the concentrations we have found out what will be the maximum absorption in the UV visible region, and so all those lambda max is documented, and based on that, so finally we have calculated the K value, that is binding constant. So from the binding constant we can understand that what will be the effectiveness of the complex. So to find out the binding constant, we have so used the formula. So that is so initially we have to draw the graph of so DNA versus epsilon A minus epsilon F, okay, into concentration. So there is a graph. We have to plot the DNA concentration versus this EA minus EF values. So from this we can so finally we can calculate. Actually this formula is not shown directly, I think. So from this we can rearrange. Actually this is the general formula, that is concentration of DNA divided by this epsilon values of free and combined form of these complexes, which is equal to the concentration of DNA divided by again so this epsilon values. So epsilon values of the next concentration and the free that is the epsilon value of free ligand plus 1 by Kb into epsilon B minus epsilon F. So this formula again we have to rearrange in order to get the Kb value. So okay, we can rearrange this value and finally we can calculate Kb. So all other parameters will be obtained either from the experiment or from the graph. So finally we can calculate this Kb value. This Kb value here it is obtained as so our Kb value is here it is 1.2 into 10 to the power 5 per molar. So in 1 mole of the solution, this much amount of that is the binding constant is this much, and it is increasing up to 4.4 into 10 to the power 5. So actually this as the binding constant increases, this anti-cancer property also will be increasing. And also from the binding constant we can understand that so whether the complex is interacting with the DNA of the cancer affected cell, whether it is through the external binding or the group binding or the intercalative type of binding. So if it is external binding, then the complex may not be that much effective. If it is a group binding, somehow which is effective, and if it is intercalative type of bonding, then which will be highly effective against the fighting of anti-cancer cells. And at the same time, this the tenfolds value also we can understand that so what will be the effectiveness of the compound. So here the generally used anti-cancer, there are so many anti-cancer drugs which is presently existing in the market. Among the cisplatin is the most common, and that is the first complex which is proved as anti-cancer, proved as are having anti-cancer properties. And the typical anti-cancer compound is ethidium complex, we used to say, and which will be having this value as 10 to the power 7, but it is that is very risky to use, which is having so much of high side effects. So that's what, in that way we can rule out those type of compounds. And so based on the effectiveness of this cisplatin complex, actually the cisplatin complexes which is undergoing the square planar type of structures, but this ruthenium complexes is having octahedral structure, and because of this that is the structural nature, and at the same time the effective K value which finds more applications in the anti-cancer agents. And at the same time, from the UV studies we can understand that so this type of complexes are undergoing hypochromism. So any complex, if it undergoes a hypochromism, generally which will be, so during the UV spectral titration, when we increase the concentration of DNA, so we can understand that so those complexes will be undergoing intercalative type of bonding. So if it is intercalative type of bonding, means these complexes can readily react with the DNA, and it can able to cleave the DNA. Once the DNA is cleaved, it cannot able to regenerate its properties, so that which will not multiply further. In that way, if we use these complexes, so this DNA will be made as inactive. So that's what the biological characterization. So as a chemist, we have found the results only this level, and maybe the next level biological peoples or the pharmaceutical or medical people, they have to take it in the next level. And in that way, since we have received a grant for this filed patent, so now there is a discussion with the pharma industry, and definitely we will be having the collaborative research with the pharma industry. And finally we claimed by this patent, so this chief bases, the ruthenium complexes, so okay, the various substitutions in which we have used to synthesize this complexes. And finally we have claimed it's that is a medicament for the treatment and or prevention of cancer, that is its applications. So these are the claims we have made. So that's what the characterization of metal complexes. So that's what, to understand fully, I have chosen one case study, accordingly I have explained. Now let me continue with the presentation. So that's what all about. Till now I was explaining about this case study. And next is so this is what the patent publication of the same complex. So we have filed in the year 2015, and after a thorough examination and so much of explanations, it's actually from this you can understand that so this is what the various documents we have submitted at the patent office. So this many documents we have submitted. So right from, so we have started in the year 2015 and up to 2021 we have defended, and keep on we are giving the explanation and whatever the controller of examination asks from the patent office, and finally we received the grant. So this is what the granted certificate for the same complex, novel mixed ligand ruthenium 3 complexes, its production method and use in cancer therapy. So that's what, when we talk about the patenting, so we must understand what is intellectual property, why we have to patent, and what is its importance. So intellectual property. Generally, properties can be classified into two types. One is physical property, that is a tangible property. The other one is a intangible property. The best example for a tangible property, physical property, to understand the physical property, say for example, a house, a two-wheeler, a four-wheeler. So if we buy a house, even after paying the money worth for it, whether we can say that it's my house? Actually, no. So when we register on your name only, you can say that it's my property. So for that, what we used to do, we have to go to the registrar office and we have to submit the necessary documents, and we have to sign, and we have to get it registered with our name. Similarly, if you buy a two-wheeler or four-wheeler, so after paying the money worth for it, we have to go to the RTO office. There we have to register on our name. Similarly, if we find out some intellectual property, so all our inventions, all our projects, if it have novelty and inventiveness, then you can say that it's intellectual property. That intellectual property, to own the intellectual property, like physical properties, we can own the intellectual property also by submitting all the necessary disclosures at the patent office. So how we are registering the physical properties, either at the registrar office or the RTO office, in the same way we can register the intellectual property by submitting the necessary disclosures at the patent office. So there we can register or we can own with our name after the thorough examinations. So this is what the various components of intellectual properties. Intellectual properties starts from industrial designs, patents, trademarks, trade secrets, GIs, layout designs of semiconductor ICs, plant varieties, and farmers' rights. So among the various intellectual properties, in addition to this, we have copyrights and related rights. So among the components, the important component of intellectual property right for an academician is the industrial designs, patents, and of course copyright. So even among these three, this patent is most important, and it is very difficult to get the grant. But this industrial designs, very easily we can get it, even the copyrights. Next one is, so from the simple example we can understand that why we have to own the intellectual property. So everybody might be used this ring pull technology to open the beverage bottles or cans. So nowadays almost all the companies are using the same technology to open the beverage bottles. So everyone might be used. So this ring pull technology, so after inventing this ring pull technology, the person cleverly he filed the patent and he protected with his name. So for that the person is getting more than 1 crore as a royalty per day. So but we people, we are all hard worker, of course, there is no doubt. So at the maximum what we are doing is, we are doing very high-fi research, even some standard research we are doing. At the maximum we are trying to publish in a very standard or a hi-fi journal having very high impact factor, that's all. So we don't bother about the protecting of the invention, the owning of the invention. So this person, so actually this technology is very simple. So for this technology, do you think that that is hi-fi knowledge is required, and the person should have a very high skill or talent? It is not. It's very simple technology, and for the technology, since he has protected and he is getting this much royalty. So that's what the importance of protecting the inventions or the intellectual properties with our name. So let me explain briefly about the patents. So why we have to get the patents, or what is patent? It is a statutory right to the inventor or the applicant by the government for his invention, which is either a process or product. So for what we can get the patent, either for a process or for the product. It should be a limited period of time. So even if we receive the grant for the patent for our invention, we can protect only up to 20 years. That too, it starts from the date of filing and not from the date of grant. For the same patent, since we have started in the year 2015, we received the grant in the year only 2021. But nowadays, earlier the process was very slow. Nowadays, that is even within 1 year also we can able to get. So in that much, there is at the patent office, and now they are doing the examination in a faster manner. So one patent I have filed in the year 2020. Now I have received the reply. So now the process is speed up, and it is granted in lieu of the sufficient disclosure to the patent office. So after submitting all the necessary documents only, we will be getting the grant, and it is only territorial right. Territorial right means if we file a patent in the Indian Patent Office, we can able to protect only within India. So otherwise, if you feel that your product may be having a very high value at abroad countries, so we can protect in the abroad countries also by the way of filing the PCT applications, Patent Cooperation Treaty applications, and which will be maintained by WIPO, so World Intellectual Property Organizations. And it is not necessary that we have to protect worldwide. Even if you feel that which will be valued only in few countries, even you can protect in few countries also by applying the same way PCT applications. And always, so before filing a patent, so we have to check the conditions, that is novelty, inventiveness, and industrial applications. So these are the three criteria are the conditions, and during the examination they will be looking for these conditions. If our invention satisfies these conditions, then we will be getting a grant. So what is novel? We can understand that it should be new, it should not be existing in any of the prior art. So inventiveness, either it should have some technical advancements, or it should have some economic significance, or it may be both. It simply does not make the invention obvious to a person skilled in the art. So anybody, if they come across, they should not feel that so what is the same kind of invention somewhere I have met, I have seen, I have used, like they should not feel they should not feel that it's copying of something. And industrial applications everybody can understand. So among the three criteria, it is very easy to fulfill the third criteria. So whatever may be the process or product, definitely which will have some values, industrial values, applications. And somehow the novelty, the first criteria also you can prove. And it is very difficult to prove the second criteria called inventiveness. And most of the patent applications are getting abandoned because of not satisfying the second conditions. And these are all the patent rights we have. I think almost we are running short of time. So with this I'll stop. So these are the various patent sites. So from the sites only we have to start. Based on the prior art search only, we can come to the conclusion that whether we can finish our research or we have to extend. So that's all. Thank you so much. Thank you for your listening, patience listening.
Thank you, ma'am. Thank you for your enlightening presentation, ma'am. According to me, crystallization, biological characterization, anti-cancer topic was mind-blowing, ma'am. In vitro evaluation, therapeutic effect, and cytotoxic effect, DNA binding was really very interesting. As we are not that much aware of this topic, as you said before, but your presentation filled our brains with innovative information about anti-cancer, ma'am. Once again, thank you, ma'am. We are most grateful to have you here, ma'am. And
okay,
also,
thank you so much.
And also congratulations for your patent grant, ma'am. And I too have filled for patent for one of my research paper. So your key points will be very much helpful for me to proceed further, ma'am. Thank you so much.
Sure, ma'am. Sure. Anything, any help do you need in future? Also, I am ready to help you.
Yes, ma'am. Thank you, ma'am. I'll note your number and email ID, ma'am. Thank you so much.
Sure, ma'am. So that's what many of the people are not aware about this topic. You are not working, especially in this topic. I have not touched in depth. So that's what I have given the that is overall presentation.
Thank you so much, ma'am. Now the session is open. Floor is open for discussion with Dr. Putribai, ma'am. Any questions?
Yeah, it's welcome. Any of you have any clarifications? I'm ready to clarify. Actually, in vivo only, which will be more reliable and it is confirmatory, but we have to get the ethical clearance, ma'am. Either by using the rats, mouse, only we can do, by animal study only we can do the in vivo studies. But because of non-availability of this facility, we are going for in vitro studies. But in vivo studies only, which is more reliable. Yeah, ma'am.